Hello and welcome to another general botany lecture video. This time we'll be talking about angiosperms, and really I just had to split this whole topic into two videos, part 1 and part 2, but everything I'll be talking about this week has to do with angiosperms, which are the flowering plants. Now, sorry, by the way, that you can't see my face, or maybe it's a nice relief for once. I just, my laptop stopped working, so I have to figure out a new way to do this. We figured it out, so let's get started. Here are the learning goals. We're going to learn the life cycle of angiosperms and compare it to some evolutionary ancestors. We're going to understand the development from an ovule to a seed in angiosperms, and be able to compare it with a gymnosperm seed. And also to understand the process of double fertilization and its two products. We will talk about what that means. Oops. Here's a list of your terms. Feel free to pause it and copy these down. Okay, so the Cretaceous epoch is the time period that angiosperm evolution most likely took place in. During this time, we had warm weather, there were high sea levels, and so there were inland seas that were very shallow. Now, the very end of the Cretaceous is when there was a large meteor impact that affected most of the of animal life, but the angiosperms snuck by and survived that. And so there is some evidence, a very controversial claim of very early angiosperm fossils from the Jurassic before about 150 million years ago. But the first kind of confirmed angiosperm fossil that's not debated is about 144 million years ago. And this might be what the scene looked like back then. Dinosaurs roaming the area, lots of insects, those had evolved, and they were quite large. In fact, insects are a big part of the story of angiosperm evolution. Here is some evidence of some early angiosperm evolution. These might be some of the reproductive structures that were kind of starting to become more specialized in between the ancestors of gymnosperms and angiosperms. So really, these are kind of these sort of structures over here that we're seeing. These are probably highly modified strobili, actually, and they've kind of become unisexual, possibly. And let's move on. So angiosperms, we call these the flowering plants, but really that's only half the story. Angiosperms are also known as the fruiting plants because it turns out everything that is a fruit was at one point a flower, and everything that is a flower will at one point become a fruit. At least part of it will become a fruit. Even this lily you see in the picture down below. Angiosperms have an extremely massive amount of morphological diversity. We see very tiny little plants, smaller than mosses, all the way up to organisms that rival the sizes of our giant redwoods and sequoias in the angiosperms, like the eucalyptus is a notable example of a particularly large angiosperm. And there are about 350,000 at least known species of angiosperms, probably at least double that of ones that are unknown. And one of the reasons why they're so successful is because they co-evolved with animals, namely animals moving their pollen grains around. So gymnosperms and all the previous gymnosperms were dependent on moving their pollen grains with the wind. The wind is abiotic; it has no preferences, and it just, it's all chance when it's wind. But animals can have preferences, and so angiosperms were able to bank on those preferences. And we'll be talking about that. Okay, another thing that all angiosperms have is seeds inside of fruits, and this has to do with that root angio. So the suffix sperm, it literally means seed, and angio means covered. So covered seed, as opposed to gymnosperm. Gymno means naked. So gymnosperm, naked seed. There you go. The difference here is that angiosperms are covered in a fruit. Those seeds, they're surrounded by a fruit. Okay, and then we have an incredible amount of diversity. Let's talk about it. Even though we do have a lot of diversity, interestingly, we see the same parts on all of the angiosperms. They're all going to have these reproductive parts, namely male and female reproductive parts. So this is kind of similar to what we've seen in previous plants, in that we've got male and female parts here. The female parts that we're looking at is sort of this whole central part that sort of goes in between. We can't quite see it there. But the top part is the stigma, and then here's this top-down view of that. The style is this elongate part. And it actually goes all the way down here, but we can't quite see it yet because all of these stamens are still attached. And that's what these are. So these large structures, these are the pollen-producing structures. And the stamens. The stamen is the word for the male parts. Okay, so let's clear up some of these terms. Stamen does mean the male parts, but the anther is the part of the stamen where pollen is produced. Okay, the filament is just this sort of the stalk of the anther that gets the pollen out there into space. And so let's just start by talking about the male reproductive parts real quick first. Actually, let's do this first. The female reproductive part is just, it's central. It's in between all of those stamens, right? Okay, and we've kind of got a top, the stigma. We've got this long part. Well, sometimes it's long, at least in this picture it is. We've got a style that's sort of in between the stigma and the ovary. The ovary is where all of the magic happens. This is down here at the base of our female reproductive parts. So now this whole entire structure, the stigma, the style, and the ovary, together it's called a pistil, okay? But we also have this term carpel, all right? And it's actually both things. The truth is that it's one pistil and it's three carpels because we have three fused carpels here. And you can kind of see that in this picture down here, right here. So in this picture down here, we've taken a cross-section through a tiny little slice of that ovary. We've turned it on its side, and we notice that there's sort of these three sections. In each of the three sections, we have a pair of ovules, at least in that little tiny slice that we took right here. Really, this whole thing is probably stacked with these ovules. Cool. Well, the ovules are where fertilization is going to take place eventually. We're going to talk about ovule development. But it's the very top of this pistil up here. That is where the pollen grains land. Okay. Moving on. Here I've taken a cross-section through a flower bud. So it's a similar type of thing. I've taken like a little tiny section, like let's say through that little flower bud right there. And this happens to be a flower bud of an actual a flower called a lily. You might have heard of it. And if not, you can go to Safeway or any grocery store and go see a lily. Careful, the pollen stains. They usually smell good, but not always. Okay, so in the very center, we've got the female parts. This is the ovary, because in theory we're just kind of looking at a slice of a very young flower bud, and so we've got the underdeveloped, immature ovules inside of an ovary. That's where ovules live. Okay, the next layer outside of the ovary, this whole kind of part right here, sort of all this, outside of the ovary, all of this here, this is the male part. Or these are the male parts. Okay, so it's these little circles. These are the Microsporangia that are forming pollen grains. So let's get into those nitty-gritty details. So here I've zoomed in on one of those Microsporangia. This is another word for pollen sac. And really where I'm looking, if we were to be looking at sort of a more developed, like an older structure that's developing the pollen grains, it would look like this. Remember, this was our stamen, namely the very top part here, the anther, is what we're looking at here. An anther usually has 4 sacs. Those pollen sacs in an anther, those are just another word for a microsporangium, and that's what we're looking at here. This is a microsporangium, which is a pollen sac. And then in here we've got our pollen grains. Okay. Well, pollen grain is a microspore, right? So remember in the gymnosperms and in the seed-free vascular plants, we had microsporocytes that were undergoing meiosis and producing microspores. Well, we actually have the exact same thing here. Really, it's no different. In fact, our microspore will undergo our microspore will undergo mitosis and form a microgametophyte. It's just that the microgametophyte is extremely reduced in the angiosperms. It's literally just 2 cells. The male gametophyte or the microgametophyte, it's just the tube cell and the generative cell. Now, those are surrounded by this sporopollenin wall, and yeah, that's it. It's highly reduced. That's the whole gametophyte. It's the 2 cells. It's wild. So this upper picture, this is a sort of a younger anther, and then down here, this is an older anther. That's in theory the same anther that's just older. So those microsporocytes, of which there were a bunch inside of this microsporangium, they will go through meiosis, as it says here, and the 4 myospores are the microspores, which are pollen grains. Okay, the pollen grains have those 2 microgametophyte cells inside. So that's the relationship there. And we're again, just to remind you from this picture, we're talking about those anthers within a flower. Oops, and then there's that. So what's going to happen eventually is one of those little pollen grains, one of these, is going to go like this, and it's going to grow a pollen tube eventually once it comes into contact with a stigma on another flower. So we're going to review all of those terms again, don't worry, especially stigma. I know that's new. Sorry, that was supposed to say shapes and sizes, varieties of shapes and size. Unfortunately, the color here is not natural. These are colorized images, but the textures, shapes, sizes, that's all real. In fact, check out that little thing right there. That's even a very tiny pollen grain, and these are to scale. Okay, so now let's move on to the female part of the reproductive system. The ovary is the most important part, okay? So remember, the ovary is at the bottom of our pistil. So this whole structure here, this is the very center of one of those flowers. It literally is right in the center. And the very bottom of our pistil is the ovary, right down here. That is O for ovary. And inside of that ovary, we have many ovules. Okay. Well, here are some of them. So again, I've kind of taken a tiny tiny slice cross section through the ovary, turned it on its side, and that's what we're looking at here. So we've got 3 carpels. There's 1, 2, 3 carpels. And again, we're going to have lots of ovules stacked on top of one another, but since this is just a tiny slice through one of them, all we're seeing is sort of, like 1 ovule there, and actually a pair of ovules here, and in theory, there's sort of 6 ovules in this slice. Okay. So at this point, everything is sort of still diploid. All right, this is a young ovary with young ovules, and we're going to talk about ovule development. So the very outside of our ovary, the outside of our ovule, all of this stuff out here, this is the ovary wall. And spoiler alert, this ovary wall is what's going to become the fruit. Yeah, delicious. Okay, so here's the young, very, very young ovule. Young. First, I wanted to tell you we've got some of the similar terms. We've got that micropyle. It's that space here that's going to allow pollen to enter. We've got that nucellus. Remember, that's the same term as what we saw in the gymnosperms. And nucellus is literally just another word for the megasporangium. It's just the set of cells that produces the megasporocyte. And remember, the megasporocyte, this is the cell that's diploid that is going to undergo meiosis. Okay. Well, outside of the megasporocyte, there's the nucellus. Outside of the nucellus, we have integument, just like we did in the gymnosperms, but this time we sort of have a double layer of integument. So that's another one of those angiosperm synapomorphies. Double integument layer. Okay, so let's look at what's going on here in a diagrammatic form. Like I said, meiosis is occurring there. So again, let's kind of break this down. The Megasporocyte, it starts off as diploid, of course, since it's going to undergo meiosis. Around that is the Nucellus. This is another word for Megasporangium. And then we've got that double integument. Okay, this sort of the arm holding all of this. It's known as the Funiculus. I'm not going to require you to know that word, but it's just a fun botanical word. And it's nice because it allows orientation. Allows you to orient yourself with this picture here, at least. Here's that Funiculus again. That word locule, by the way, that just sort of means the space that you find an ovule in. Okay, so we've got our Megasporocyte in the middle there. This is a real image of what we were just looking at in the diagrammatic form with all of the associated terms. And then meiosis occurs. Now, we've got 4 haploid megaspores. All right. And here's what they actually look like. This is a real image through a slice of one of those ovules that had just undergone meiosis. Pretty cool. Well, pretty quickly, 3 of them abort. One of them is known as the functional megaspore. This is of course haploid because it's a meiospore, the result of meiosis. That one functional megaspore is going to undergo mitosis, okay? And that's what's going to form the gametophyte. And that's actually the exact, that sentence. I could have said that in light of gymnosperms, in light of seed-free vascular plants. The megaspore undergoes mitosis and it forms a megagametophyte. Cool. But in the angiosperms, the Megagametophyte is pretty unique. It is known as an embryo sac. So let's talk about this crazy embryo sac thing. So again, the Megagametophyte, it's literally also known as an embryo sac. Same term. It's inside of a megaspore. The megaspore never sort of releases the Megagametophyte. It stays closed. Just like everything here really does. The Megasporangium stayed closed and everything kind of staying attached to itself, it seems like. Okay, the megaspore was inside of the Megasporangium and Nucellus is inside of the integuments, and all of this together is what an ovule is, okay? Okay, so let's look at this Megagametophyte, the embryo sac, which really, it's this structure right here. And, this is a nice real image of it. But it's kind of complicated, actually. Like I said, it starts off from just this one functional surviving megaspore will undergo mitosis a few times and it'll produce these 7 cells. But one of those cells has 2 nuclei. We have these 2 polar nuclei sort of in this middle of this large central cell, which is sort of everything like right here, okay? And then you really don't need to know the fancy names for all of these terms except for egg. I mean, that's not a fancy new term. An egg is just that haploid female cell that's going to undergo fertilization soon. The antipodals, the synergids, those are the other 7 cells that make up an embryo sac. And so they're always in diagrams and everything, and I don't want you to think you have to kind of memorize those. It's good to know that they exist. The most important thing here to know is that we've got an embryo sac is composed of 7 haploid cells, and one of those cells, it's not exactly haploid, right? Again, this central cell right here, it has 2 haploid nuclei, right? There's one right there, and then there's one right there. So it's wild. It's actually, it's really not haploid or diploid, since they're not fused together. They're this other kind of strange thing called dikaryotic, which is wild. Don't need to know that word yet. But something crazy is going to happen there later, so just hang tight with that. We know the egg cell is going to undergo fertilization soon. Okay. So here's a nice angiosperm life cycle chart. I like this one. It's kind of simplified, sort of, compared to another one that I'm going to show you in a sec. And it's got what we just talked about. First, we've got the megaspore. This is still in the sort of the blue diploid side here on the left. That Megasporocyte undergoes meiosis, and one of the 4 meiospores will survive, okay? That one is going to undergo mitosis, so that's what I like about this diagram is notice that one surviving megaspore is gray, and then here the embryo sac, too, is gray. And that's because it literally is derived from that one surviving megaspore. Okay, the Megagametophyte. All 7 of those cells, all 8 of those nuclei are derived from that one megaspore that survived. Cool. Great. Well, we've got to have the pollen somehow arrive in that micropile to get close to that egg before we can have fertilization take place. Oh, here's another slide. This is a more complicated one. It's just more detailed, really. It's not that much more complicated, I shouldn't say. It is a summary of what we've been talking about so far, but just in a nice diagrammatic order, so please feel free to pause this. Study it. Connect it to what we talked about earlier. And we're going to kind of go over what happens after fertilization here pretty soon. Okay, so we've got 2 processes that happen separately in the angiosperms. We've got pollination, and then we've got fertilization. Pollination is when we've got pollen grains finding their way from an anther to a stigma up there. Okay, fertilization is the same thing that we've talked about. Fertilization is when a sperm cell and an egg cell fuse and form a zygote. And the pollination and fertilization won't happen at the exact same time. Here's a really cool close-up look at a scanning electron image of pollination. We've got pollen grains here that are on the tip of a stigma. So this is a 5-parted stigma. Remember, the stigma is just the top of that female reproductive part. And that's where it's receptive to pollen grains. Okay? So it's these little yellow pollen grains that will, from here, be able to germinate a pollen tube all the way down this style. And then, in theory, all the way down here to the ovary, which is, in theory, down there somewhere with ovules inside of it. Cool. Fertilization is this part that I just drew a second ago. Fertilization is the part where we've got a pollen tube that grows and it finds an egg. Okay. This is a cool image of a whole bunch of these pollen grains that have each germinated a pollen tube, right? And fertilized these different ovules. These are all different ovules here on the left. And so all these are pollen tubes. Super cool. Okay, so we have something wild going on here. Double fertilization. What happens is the Microgametophyte inside of that pollen grain, which, remember, it was just those 2 cells. It was the tube cell and the generative cell. Well, the generative cell is going to undergo mitosis and form 2 sperm cells. And here we go. This is what that looks like. The tube cell nucleus it's going down because we have already germinated this pollen tube. That's what this whole long structure is, a pollen tube from our original pollen grain right here. And then the other cell here, the generative cell, which was originally just 1 cell in the pollen grain, it has undergone mitosis. It is now 2 sperm cells. Wild. Crazy. Well, one of those sperm cells is going to fertilize the egg cell as expected, and the other sperm cell is going to fertilize those 2 polar nuclei in the center of that embryo sac. That's wild, because those, remember, those were this weird sort of ploidy. So this is what I mean by double fertilization. This is something that's really special to the angiosperms. And now that we've got this sort of other fertilization that took place, not just the egg cell, we kind of have this other tissue that can potentially be nutritious to the embryo. So again, those two sperm cells, 1, 2, one of them is going to fuse with the egg cell, fertilize and form a zygote. The other one is going to fuse with those two polar nuclei in the middle of the embryo sac and form this triple nucleus fusion. And so it's going to be something that's literally called triploid, 3N. This is wild. And triploid cells, they don't really work very well. And so this isn't going to go very far. It can be nutritious for the embryo, but it's not going to be able to form like a sporophyte or anything. Kind of have even numbers of chromosomes if you want kind of good meiosis. And okay. So here's the double fertilization and the results. Okay. There it is. We've got that triploid tissue. And now originally it's just this one cell. It's called the endosperm mother cell. And then we know the other side here was a zygote. And then those are going to undergo mitosis. And now we've got the zygote is going to become, frozen. An embryo. Yeah, the zygote underwent mitosis to become the embryo. And the endosperm mother cell, it was just a single cell, and it underwent mitosis to become this tissue called endosperm. Okay, let's talk about what endosperm is. Look at pictures of it. Oh, and this is what a seed is, by the way. Kind of a big deal. Right, so an ovule, everything we talked about earlier, with the megaspore and the Megagametophyte and the embryo sac. Well, that was a baby seed. An ovule becomes a seed after it has been fertilized. That is the relationship between those two terms, ovule and seed. Okay, so the ovule and the seed, they're both inside of the seed coat. Just a reminder of what we talked about earlier. All of those layers. And what we had earlier, that embryo sac, remember with all of those interesting cell types, including the egg cell and the polar nuclei, those have undergone double fertilization, and the egg cell has become an embryo. And here the embryo is really kind of actually all of this sort of darkish, purplish tissue here. There's its two leaves that I'm circling right now. And yeah, that's the embryo. Yeah. And then the endosperm is this light blue stuff on the outside. Yeah. It's the nutritious stuff. Okay. And here's the middle area. So we can really see the endosperm. It's most of this stuff kind of originally. Remember, this is kind of just one cell for a second. Once that one sperm cell has fused with those two polar nuclei. And then it undergoes mitosis. And it forms all of this stuff that's sort of in white that I'm now coloring in red. All of the stuff around the embryo inside of the seed is endosperm, and it is nutritious, protective, and it helps the young embryo develop inside of the seed. Okay. All right. So just to bring us back to where we were talking about earlier, we're all talking about this. This is all happening inside of a flower. Our next video, we're going to talk about really the fun parts of a flower and everything. But the ovule and the seeds, these are developing inside of the ovary here, okay? And so the ovary is going to develop just like the ovules were developing. So the ovule, here's a sort of an image of an embryo developing. All right, and the ovary also develops, okay? The ovary becomes the fruit. So this is one of the things that make angiosperms so special. That whole story of the ovule and the endosperm, that happens inside of a fruit. So it's even more protective. It's got even more potential nutrition if it needs it. Okay, so in a coconut, for example, a coconut is a fruit. An entire coconut has this kind of papery, well, not papery, it's like fibrous, hairy stuff all on the outside of what you might think of as like a typical coconut, which is actually just a seed. And the sort of milk of coconuts is actually liquid endosperm, that triploid tissue. And then the flakes of coconut, that's the same thing. It's more endosperm. The embryo is just sort of a little structure. You probably wouldn't even notice it. And then all of this other fibrous stuff. You can look up a picture of this if you want. You can Google it or something if you've never seen a full coconut. I know I hadn't really before learning about this. That's the fruit wall. Yeah, that's the ovary wall. All of this stuff outside of that seed. Okay. That literally, what I'm saying is this was literally a small flower at one point. That was the ovary of a small flower. And even like this corn kernel. Think of a corn as a kernel. Well, it's actually a fruit because a fruit is a matured ovary. And here we have an embryo. Right? We actually have those words we learned last week. A radicle, that's another word for a root tip. And then up here we've got, we haven't learned those words. You don't need to know these words, by the way. They weren't on your list. These are those embryonic leaves. And then this is the embryonic root. So this is the embryo, in other words. And then pericarp here, this is the actual fruit wall surrounding the embryo. So a corn is literally a fruit. Cool. So is a coconut. Oh, and importantly, comparing the angiosperms with the gymnosperms, okay? Gymnosperms, they did not have endosperm. Instead, this tissue sort of around the embryo was the gametophyte. And their seeds are just sort of naked. They've got this seed coat, but they're just sort of attached to a cone scale, right? They're not surrounded by ovary wall. Okay, so again, a little summary here. The ovule becomes the seed. Okay, within the seed, you've got the embryo and the endosperm. And the ovary of the flower is what becomes the fruit. The seeds are inside of the fruit. So when you think of your typical apple, maybe you've known this, maybe not. You go to the store, you buy your apple. That's great and delicious and all. But what an apple is just an old flower, namely the ovary of that flower. So if you watch the development of an apple, it would start off as a flower, and then this base of the flower here, it would start to swell up, and then this flower, it would lose these petals. They would just fall off because they're no longer needed, and then it would develop into this juicy, delicious apple with seeds inside. Cool. And then those seeds, once, hopefully it'll somehow find the ground from the plant's perspective. And there's many ways to do that. We'll talk about seed dispersal in the next video. And then the seeds will form primary growth. And remember, way back in the beginning of the semester, we talked about sort of primary growth in shoots and roots. It definitely won't, remind you of many of those terms. Just remember that they exist. There's a root apical meristem and a shoot apical meristem. And those form really in the embryo. And those are the very kind of first noticeable parts of a young sporophyte. Yeah. We also did learn this term last time in the gymnosperm video. The term hypocotyl, that is a term that refers to sort of the space in between the very first root and the very first shoot. So hypocotyl is neither a root nor a shoot. It's sort of that space in the middle. Interesting. And that's it. See you at the next video.